thank you all. I have been enjoying this, and I hope that you will like molecular biology a little bit more by the end of my class, of my classes. And I was a little bit optimistic, so I have this class prepared of stem cells and IPS, and also the aging class. But if we don't manage to reach it, you have the slides, so if it's something that interests you, you can go through the slides. And the aging part is mainly what we do in my lab. So if I manage, I try to tell you a little bit about the research that we do. But again, to focus on epigenetic importance, we will talk today about stem cells and IPS. And just a reminder to put everyone on the same page, the stem cells are cells that from them we can generate every different tissue in the body and in any organism. They can self-renew indefinitely. They can divide and divide and divide without being cancerous cells. And they have the ability to differentiate in any type of cell. So we have different types of stem cells. We have the totipotent stem cells. And these are the ones that no one usually talk about. Most people talk about pluripotent stem cells because totipotent is one layer before. And these totipotent stem cells can also form the placenta. So when we are generating from a zygote embryo, we also generate the placenta. So totipotent stem cells can produce placenta. Then we go to the next level, that is pluripotent stem cells. And these can generate every organ in the body. Then we go to multipotent stem cells that they can generate a specific line. For example, if we are in the blood, so every blood cell. And then unipotent, they have the capacity to renew just into one type of cells. So how this happens, we have the fertilization, the first divisions, and then we go to the blastocyst, and from here we generate an embryo and a full human, rat, mouse, whatever it is. So the cells that we produce to produce stem cells are from here, from this part. So again, that's why we get pluripotent stem cells and not totipotent stem cells. So what is the difference and how can we recognize a stem cell? First of all, the self-renewal. They will divide and generate more of themselves. It can divide and generate more of themselves. And in this way make copies and produce as many cells as we need, and also to keep a tissue healthy if we need this tissue. And they will differentiate into specialized cells, nerve cells, muscle, whatever is needed for the differentiation. And we can have, as we were saying before, symmetric divisions. So we have from one stem cell two new stem cells, and this keeps us the pool intact, and we generate more stem cells. So usually this happens when before differentiation, that we want to generate a lot of stem cells, and then we start differentiating the cells in some cases. In some cases we have two cells that differentiate, and if we want to keep the pool, one cell will still be stem cell and the other one will differentiate. In this case, every stem cell that we use, we didn't lose our pool, right? Because we always remain with one. Okay, so if we have a stem cell that differentiates into a specialized cell but renews itself, from these divisions we can get 4 specialized cells and still remain with one stem cell. And in this way we don't deplete our pool of stem cells over time. In some cases we get all specialized cells and then we lost our self-renewal capacity. So we have in our bodies 3 types of stem cells: the embryonic ones that originate from the blastocyst, and they have, as we said, pluripotent capacity. Yeah.
Well, just a quick question. Why 4 specifically?
No, just here in this example, it was 4 cell divisions, but they can go through many, many cycles, but just as an example. So in here, we have, at least in humans, 254 different cell types in an adult tissue. So all of these cells can be generated from the same pool of stem cells. Then we have adult stem cells that usually they are not pluripotent anymore, they are multipotent. They can generate a specific line because during the differentiation and the development they already acquire some characteristics that are unique. And they cannot jump between one and the other. They can do small jumps into the differentiated state. But they also can divide and renew themselves, and they help maintain the tissue. That yesterday there was a question, right? How, if the cells cannot divide forever, how we can maintain them? Because we have these stem cells. But they can get damaged, and we'll get there. So we start losing our pool. And last, we have a new kind of synthetic stem cells that are the induced pluripotent stem cells. That they were not stem cells, but we synthetically generated them in order also to do research and hopefully for the clinics. So we can take adult cells and reverse the process of differentiation and get new induced pluripotent stem cells. So we'll go through all of this and the process to produce them. So first, again, we have this scheme where we take the blastocyst that is a really, really early embryo. It's not even morphologically an embryo. It's a sac with many, many cells. And I think this is important to say because many people oppose the research on stem cells and taking this kind of cells, particularly from humans, because they consider it an embryo already. And I don't think this is an embryo, but it's open to debate. And this debate also led to the technology of IPS, because in the United States it was completely banned to use stem cells for many, many years. And then people came with ideas of what we can do, and all this new technology of IPS was invented or developed by Yamanaka. And then we have our different tissues that are already adult tissue. So these embryonic stem cells are from the inner cell mass, and this outer layer is the trophoectoderm. So again, this layer will form the placenta tissue, and since we are taking the cells from here, we are losing this differentiated layer, and we are losing the program to produce totipotent cells. So we don't really know how to produce them yet, but there are many labs trying to develop this also to have the capacity to grow embryos outside when it's necessary. For example, when it is necessary, if there is some problem, or also if, for example, in the future, one of the technologies that we want to develop when we are losing a species is if we have the DNA and we have a sperm and we conserve sperm from a species, we still need to grow these embryos somewhere, right? But if we develop the technology to produce this outside the cell, then we can maybe revive some species that were lost, maybe like a Jurassic Park kind of thing, but not the dinosaurs. So this is also an important technology, but we are not there yet. We don't know how to do that. And then we take these cells and put them in a tissue culture. Any of you have grown cells in the lab? Yeah? Okay. So it's like a pet, like a, I don't know, for your generation, it's a Tamagotchi. You have to come and feed them, and if you don't look at them, they get contaminated and they differentiate, and so they are very, very particular. Specifically, stem cells, if you are constantly caring for them, they start differentiating, and the first thing they do, the default is neuronal tissue. So if you don't take care of your stem cells, they will start becoming neurons. So this is the default. It's very interesting that this is our preferred pathway to become a neuron. And then once we have these embryonic stem cells, many labs are doing research into how to differentiate them. Which kind of signals do we need for each one of them to get a tissue? For example, to get specifically dopaminergic neurons, it takes about 90 days of growing the cells, and every 2 weeks you start adding a new growth factor or a new factor that is specific for these neurons to divide into specifically dopaminergic neurons. So it's a very complicated process. It takes a long time. Another thing that you can do, and it's also very cool in the lab, is to develop cardiac tissue, heart tissue, and after about 2 weeks, they start pumping all together. You can really see this in the plate. It's really, really amazing. So you are growing the stem cells. They differentiate into hard tissue. They get connected, and they pulse. So it's really, really cool. This is like the intrinsic knowledge of the cells already to start this process. So it's very interesting, and of course we want to understand how it happens and if we can reproduce it, also to understand simply because we want to understand, but also to reproduce this in case we want to regenerate tissue, right? Most of the horrible movies are about clones that are being used to gather the tissue for the rich, right? So if we develop these methods, we don't have to take the homeless and steal their tissues for the rich people. Okay, so how this happened? We have, again, our same DNA, and then it can choose to go downhill. This is called the Waddington landscape, meaning that we start with a very open landscape, and it will go downhill. So everything, when we reach this point, for example, we cannot come in here because we have an energetic barrier to become something else. So in this case, when cells start differentiating, going downhill, for example, these are two different kinds of blood cells, B cells and macrophages. For example, from here to here, the barrier is smaller. It still doesn't happen in nature, right? But if I want to convert this one to this one, I need less energy or do less changes than if I want to go back to this pluripotent stem cell. So we start here in the top of the hill with a totipotent cell. Then it becomes a pluripotent cell. And then it can go to different cell types. And usually we get, for example, here hematopoietic stem cells will give me all the unipotent cells that can form beta cells, macrophages, microglia, etc. Or to come to a different path, for example, here, and get fibroblasts and muscles and everything else. So we go downhill, and then the energetic barrier is very difficult, but still, this is what we do when we generate iPS. We break this barrier by inducing changes in the chromatin. So there are many signals, just all the names are not important, that we have discovered over time with research about what makes a pluripotent stem cell. For example, OX, SOX, nano are some of the factors that we put to generate iPS, and some of the signaling downstream to generate the differentiated cells. So this is an ongoing investigation, and this is what, from a single genetic code, we can generate all the different tissue because we generate a different epigenetic code. We close the regions that are not important for us. We left the other regions open, but we have the same DNA, right? We started with the same DNA. We ignore recombination, rearrangements, and all these transposons that get inserted to generate variability, but more or less we have the same genome. So in the germline, we have the genetic code from both parents. In the zygote stage, we delete most of the epigenetic modifications. So we have to remove everything or almost everything in order to add the modifications again, because they come with the information of I am sperm and I am an oocyte, right? But now we have to delete these modifications and start anew to form every possible tissue starting from stem cells. So most of the modifications are deleted. We have few cases of imprinting. Imprinting are very, very few genes in the genome that remember if they came from the mother or the father. And they keep this information, and usually one is silenced and the other one is not silenced. Yes?
You say most. Yeah. You say most of the epigenetic modifications are erased at this stage, and usually when people are talking in general public talks about epigenetic, they usually talk very much about inheritance, and that it is another layer in inheritance, and we should consider this in our populations and the general. How relevant is inheritance in the epigenetic material, because if you say now that most of the epigenetic modifications are erased at this point, it wouldn't have really much of an effect.
So it's a very complicated question, because we do see that some things keep the information, but we see that the epigenetic modifications were deleted. So how they regain these same specific modifications, we don't completely understand, right? Let's imagine, like with the example I gave you, you remember with the mice that have a caring mom and not caring mom? So this caring is happening in the life of the mouse, right? But when they are born, they are already born with specific methylation or non-methylation in these genes that are for stress response. But these were deleted during the embryonic stage. So they are regained, and there are various possibilities. One of the possibilities is that still through maternal RNA and maternal signaling, we signal to bring back these chromatin remodelers back. And for example, other species, like C. elegans, they solve the problem in a different way. They can transmit information from the grandfathers to the next generation by using siRNA. So they generate siRNA that can bind to the genome, and in this way, it's not in the genome, it's above the genome, but it keeps information of what to keep open or closed. In mammals, we have tried to see if we have a similar mechanism, but it has not been found, like how you keep information from what happens in somatic tissue, or what happens, what was the original marking in the mom or the dad. And we still don't understand completely how it happens, because most of the things get erased. And just few regions get this imprinting that they keep a signal, usually methylation, reminding these genes if they are maternal or paternal genes. I think, I'm not sure if we have it in here. Okay, I didn't put the example here, but there are two diseases called the Addison Syndrome and I don't remember the other one. But what happened is that you have the maternal gene that is usually active, and the paternal gene that is usually not active. And in some people, there is, for example, a mutation in here. So what will happen is that, let's say this couple get married and they have kids. So if they have, if they inherit, and the other person is okay, let's assume it has the correct gene. So this person that got these genes, and this person of these genes got married, and they have, and this was the mom. If they have kids, the kids will be sick because this gene is expressed from the mom. No matter that here we get a correct gene, so they can get the correct gene, the one that is incorrect. This one is always silenced because it came from the father, and this one is supposed to be active, but is mutated, so this person gets sick. If it was the other way around, this was the dad, and this was the mom, then even if they get the mutation, they are healthy, because they remember that they have the gene and they are using the other copy. So this is imprinting. It is in a very few regions that we know, and it's mainly kept by methylation, and these regions are kind of guarded during this erasure period. But also the mechanism is not completely understood. We also think that may get protected by long non-coding RNAs, like a little bit similar with the siRNA, but it's still under investigation. So most of these things are deleted. Many of the signals may come from the maternal cytoplasm or from signaling that activates the production of certain chromatin remodelers.
Also in one of your previous slides, this one.
Mm-hmm.
I remembered from a previous moment when you had said that essentially differentiation will happen when we sequentially turn off many of the genes.
Yes.
Right? Why? I don't know if that will be also one of those questions without answer, but how does having all the genes active not lead the cell to stress or some sort of thing like that? Because
yes, so what happens, and we will see in a few slides, is that we have what is called bivalent domains. So usually genes can get modified epigenetically to be active or to be, let's say, repressed. And most of the genes will have one modification that is telling me get active, and the other ones get the modification do not get active, silence me. During the ESL period and in embryonic development, we keep bivalent modifications. So they can go either way. They still are like kind of deciding which one should I keep. And then they are not as active. We get a little bit of noise, but we have to have an extra signal to allow expressing these genes. So they are still undecided, and then they'll go and take the decision. And we will see also that we will change topologically the DNA to close or open the regions when we start differentiating. So this modification, together with the packaging, will give the final decision of what should be active and what should not be active, because not all of the genes are expressed, because when a stem cell is just self-renewing, it doesn't need to produce, I don't know, melatonin or serotonin, right? They don't need it. But they have the genes still in a kind of open way, sometimes with these bivalent modifications in case they need it. In case they get the correct signal, they will activate it or repress it. After many signaling and asymmetric divisions, they start deciding where to go. So this is what happens, but they are not expressing everything, no? Because this will be a complete waste of energy. But they could if they get the right signal. Okay? So after this, we start adding the new modifications in the epigenetic code and changing the topological conformation of the cell. And then we have differentiated cells. So again, if we have our totipotent cell becomes a pluripotent stem cell after one layer of differentiation or losing this capacity. And then if we go to a blood stem cell, we can produce red blood cells and white blood cells. But if I became a muscle, I will just produce this one or that one or that one. So we are losing our capacity to be everything, right? That by taking some choices, we lose this capacity. And this is one of the experiments that I was talking about. It was a very simple but elegant way to prove the chromatin becomes more compact in stem cells. So what they did is they took a fluorescent protein that is bound to the histones, for example, HP1 or some histone, H3, H2, with GFP. And what they do is they do photobleaching. So imagine this is the cell, this is the nucleus, and it is all fluorescent because it has histones everywhere. And then with a laser, let me mark it more like this. We delete because we are kind of burning the sensors with the photons, and then this part becomes not fluorescent. And then I can measure how long does it take to get, again, this stage fluorescent. The proteins that were burned, they are not fluorescent anymore. But it depends on how much movement I have of the chromatin in there, how fast I'll regain fluorescence in this site. So if I take a stem cell and do this, I get really, really fast movement of other histones and chromatin, and I renew this. I take the same stem cell, it's not a different one. I differentiate it to be a neuron, and then everything becomes slower. And then it takes me a longer time to replenish this site. So it's a very simple experiment, but in the end of the day, it's telling us a lot of information about what is happening. So in an embryonic stem cell nucleus, we have the DNA that is starting to attach to the lamina to the silence genes, and some loops that are open, but in general it's more open. And then as we start differentiating, we start closing some regions and having these topological associated domains. We start adding more of these HP1 proteins that kind of close the chromatin. The lamina is nicely formed, so we also gain less flexibility of the nucleus to migrate. And then we start adding different methylations in specific regions, not all over. And then we add the histone modifications. So now we have domains that are for active chromatin or for silenced chromatin. For example, these two are silenced chromatin, and this one is open chromatin. So you also can see that H3K27 and K methylated and K9 methylated are in more compact regions. And for example, this one that is an active promoter, it's in a more open region. And then we start organizing really the heterochromatin, putting to the periphery everything that should not be used. And in the center, the things that will be more used, and the formation of the nucleolus. So at the beginning, we are very flexible. Anything can happen. We are giving them flexibility to be whatever they want. But then we start forming the different domains, and these different domains will vary depending on the signaling that we get. I'm going to be a neuron, I'm going to be skin. What is my future, right? So in this way, we start packaging the chromatin in different ways that this allows us to use only the genes that we want to use and silence the other ones that we don't need. So in adults, we have stem cells in the eye, in the brain, a little bit in the hippocampus, in the skin, breast, the gut, testicle, muscle, bone marrow. And this is the experiment I was telling you yesterday, that how long the cells live. For example, the turnover of a cell over time. And you can see that in the gut, we have cells that live usually 2 to 4 days. And then they completely gone, and we have new cells. And if we go to the end of the curve, ovocytes, we are born already with all ovocytes that we are going to have for our life, and they just mature. But we don't divide them anymore, and we don't have new ones. In the eye, for example, lens cells also. These are the ones that we have, and that's it. Skeleton and central nervous system, there is a renewal of 10% per year, or most of our neurons is for a lifetime. Also, we have some stem cells, but the ones that we have and did connections from childhood are the ones that will carry on with us unless they die for a lifetime. And this is the experiment, or not on purpose an experiment, but this in the 60s was the bomb era testing for nuclear energy and nuclear bombs. And the concentration of label carbon-14 really changed. And then people that were born around this stage would be labeled with carbon-14, or more carbon-14 than usual population, right? And then you could start seeing the decay of the labeling. And for example, in the gut, that it changes really, really fast, so we lose all the signaling. But in the brain, it will be less lost. And in this way, they came with this dating of the how long a cell requires to renew, and for how long do they live. And this also correlates with the capacity to self-repair and have stem cells. So for example, the one that have a high turnover and have a high regenerative potential, for example, our blood. If you go and donate blood, nothing happens to you. You produce new blood, and that's it, right? We are all okay. And then as we go along this line, we have a lower cell turnover, still have some regenerative capacity, and then the cells that don't have a high turnover, we have a very low regenerative potential. So if it gets damaged, then we have a difficult time recovering from this kind of injuries or disease or things that damage these tissues. So why we lose our stem cell pool? Usually when we are young, we self-renew, and then we have cells that can keep us and keep regenerating our tissue. But as we get older, some of the cells just lose the capacity because they differentiate. they didn't get the signal to be kept as stem cell. The niche is very important, so in some cases you get wrong signals and differentiate a little bit, so we lose some of them. Or also on purpose, we differentiate some of them without asymmetric division, just differentiating divisions, and then we lose some of this pool. Some of them don't self-renew, they just lose the capacity, sometimes because of damage. If they are damaged, they don't want to become dangerous, so they don't renew anymore. Some of them become senescent. They understand that they have damage, so they don't have anymore the capacity to divide. And some of them become malignant. They didn't get the signal that something was wrong, and then they are transformed and could lead to cancer. So this is why when we got older, we don't have the same renewal capacity in our tissue that we have when we were young. So how is the tissue replaced? And for example, one of the very beautiful examples is the stem cells that we found in the bone marrow, right? Because from there we produce all of our blood cells. Then they differentiate into all the different kinds of cells that are in the body. These are multipotent stem cells, right? They cannot become a neuron, but they can become any kind of cell that is in the blood. And these are the different lineages that we can have from the hematopoietic stem cells. So we have cells that produce antibodies and cells that transport oxygen and cells that recognize in a non-specific manner all the pathogens. So we have all of these cells. And one of the things that this understanding gave us is the capacity to do bone marrow transplantation. So the first experiment in mice, what they did is they took a donor that is a mice that was expressing GFP. So we can identify the cells that is produced because they are labeled with GFP. And then the recipient was irradiated and all the bone marrow cells were killed. So now we put bone marrow from the donor, and we can see that the cells that are produced are all GFP positive, telling us that this bone marrow transplantation allow the regenerative capacity of the other mice that have no bone marrow at all, and produce all the blood cells that were needed in the new mice, right? So usually it's a little bit different between mouse and humans. In mouse, it's about 14 days, and you already have red blood cells and platelets and all neutrophils and different kind of cells. In humans, it takes about 2 months. When we do, I am sure that also in here you get all the time advertisement to come and check if you are a candidate to donate bone marrow for some patients. So if you are a candidate, when your bone marrow gets transplanted to the person that needs it, then within some days, it will start expressing within 2 months. They have their own self-renewal capacity from cells that came from you. So now they have some of your stem cells and multipotent stem cells and can produce all the red blood cells and all the hematopoietic stem cells. And of course, this is very important because it allows us to cure some disease, and also because we understand what is happening, we can help other people with this kind of technology. Another example of stem cells, and here the niche is important, are the gut stem cells from the small intestine that also have different kind of cells, Paneth cell, goblet cells, endocrine cells, and columnar cells. And in here, this is also something that I tried to draw for you the other day. We have these cells that are the dividing cells, and then they push up and start differentiating. So these ones will be differentiating. And in here, when they are done with their career, they go to apoptosis and get cleared from the system. So in here, we have a very high regenerative capacity. We said 2 to 4 days is what these cells usually live. And they start differentiating in a way that also has a gradient of factors that are expressed. So how they go from here to here, it also depends on the neighbor cells. They get signaling, also mechanical signals, and also a gradient of things that are secreted in here in this layer. And we can see that as we get further away, we will have a different concentration of these transcription factors that will influence the final development of these cells. So location and the niche around it can help us to get to the stage that we want. We have a gradient of signals that get diluted or get more concentrated, depending where they are coming from. And this combination is what gives us the final cell that is completely differentiated. And for example, in Drosophila, for those of you who studied biology, one of the typical examples is the discs they have. So they start expressing some transcription factors that are concentrated, very concentrated, coming from here, and some others are coming from here. And then you get different concentrations of each one of them and the combination of both of them. And this gives the different disc of what everything should become. So we have the combination of the different concentrations that go like this and like this. So in here we have high of this signal, and in here high of this one, and in the middle we have both of them in the middle. And these combinations of signal can give a very different outcome. Only by just two different signals, the outcome can be completely different depending on this gradient. Okay, and here is another example of stem cells that are in the hair. We have our stem cells, and we will have regenerative capacity if we kill some of the cells in the upper layers, but not if we kill the stem cells in the hair. So this is what we were saying about the niche. It's very important to keep the cells from start differentiating or dividing, and if the niche gets damaged, if we get signals that damage the niche, then we can lose our stem cell capacity because we get mixed signals that shouldn't be there. There's a question? No? Okay. So we have to maintain the adult stem cells in a quiescent state, meaning they are in G0, they are not dividing, but they are not senescent. And just when they get the signal, for example, after injury, for example, when we damage our muscles and we get a cut or whatever, we start regenerating. So we get a signal from the injury that we need new muscle cells to be produced. So we activate the stem cells to start dividing and differentiating, and usually try to keep cell renewal or not. And this can be also metabolic signals or physical signals that, and immune system signals that give the niche the information it needs to know if it continues or if it remains in G0. So what is the problem with the use of stem cells? As I told you before, human embryos, that is a complicated topic, particularly for religious people, and that they are not autologous. If I need stem cells to cure something and they are not mine, then my immune system may react against them. So it's more complicated to use. So there started the long career of cloning in mammals, and it raises also many complicated questions. So should we do it, and why, and how does it work? And if it's ethical to do that, right? There are some in the news, some rich people that clone their dogs over time or whatever it is. And it's like, okay, it's a little bit problematic. And also we see that it's not completely without damage to the cloned animal. So we have clones. What does it mean? The organism is identical to its parent or the DNA where it came from. Yeah.
So you've talked about like how a stem cell needs to be from your own body to recognize, and I believe that is due to the MHC complex, right? So my question is, when does in the development of a stem cell the transposon that forms the MHC complex works? Because then, like, if you are using a totipotent, I believe that the transposon can be used by, like, following your own body rules, and then you have the same MHC, and then it will be recognized by as a cell, you know.
So that's why we always, when there are transplantation and these kind of things, you check for compatibility, usually as closer as the genetic, for example, if it's your brother or if it's your cousin, you have higher chances that your immune system won't recognize them because you have similar proteins, even if they are not your own. The genetic code is almost half or to your parents and almost 80% of your siblings, that you get a better outcome. But this is one of the main barriers in stem cell technology, because you still can raise the immune system just by having different polymorphism, a different variant of the gene. So that's why in many cases it doesn't work. It works better in isolated places where the immune system is less accessible. For example, there were some experiments putting stem cells in the brain. So in the brain we have a more isolated immune system, and then the compatibility didn't have to be as high. Still it has to be as high as we can do, but we have like a immune system isolated region. But exactly what, if the donor is very distant genetically, it usually won't work. But I don't know if this answers the question, because also it's very difficult to understand all of you with the masks.
Yeah, but like we cannot like engineer the constant portion of the histocompatibility complex to
remove the MHC complex.
Yeah, to make two types of.
Maybe at the experimental level could be a very cool idea to do. Usually, in the ethical topic of putting stem cells, they want as less as transgenes and modification as possible. So for example, if you had a cystic fibrosis and the gene of cystic fibrosis is mutated, giving you the correct one is okay. But doing other enhancements, it really is limited by the ethic committee. So maybe also if you don't have an MHC, if you completely delete it, if the cells become cancerous or infected, you don't have a way to protect these cells. So this could also be a barrier in developing that technology. As an interesting experiment for compatibility, it may increase compatibility, but it may come with more chances of cancer, infection, and lack of control over these cells in the body.
Okay.
Yeah. So we have two main methods to clone. We can do an embryo transplant and adult cell cloning. So we will see this adult cell cloning. That was what happened with Dolly. So it was the first mammal to be cloned, and they took the genetic material from the mom, it was breast tissue, and put it in the egg of a different adult, the donor adult, and the cells start dividing and they became an embryo, and then this was implanted again in how do you say, like another sheep that was activated with estrogen and with hormones to be to pretend that she's pregnant, and then the clone was born. So we have this genetic reprogramming that from milk production, in the case of Dolly, it has to be reprogrammed to get us this nucleus, right? We are taking the nucleus out of the ovocyte and we are putting in, I'll show you a movie in a moment, the nucleus from the mammalian tissue, and it already has its epigenetic modifications, its everything. So when it comes here, it gets all the signals from this cytoplasm, and in some cases, and you see the efficiency, it works, but also the survival is very low, and also very few survive to birth, and they have many abnormalities. So maybe the DNA is okay, but still also remember that if it's tissue that has been going on for a while, we have maybe also accumulated damage and have mutated genes. So also we have the reprogramming barrier and also the damage that may already happen to these cells. So this is the efficiency. For example, if we created this number of embryos per per species, for example, 7,000. By the end of the experiment, at the time of the publication, they have very, very few left, very few live birds, and not all of them develop from the blastocyst to be transferred. Embryo transfer is to already be implanted in the mother, in the new mother. So it is very inefficient. It has a lot of ethical issues, and also the progeny, the animals, are not completely healthy. And it's because, of course, we don't erase all of the signals, and we accumulated mutations that cannot be erased. So how can we do this reprogramming? And for this, we can increase the rates of success if we play with epigenetic code. For example, if we take the same experiment, but then we add the acetylase inhibitors that remove histone modification, we prevent the removal of histone modifications, and then we have an increase from the control in the rates of success. And for example, a kinase inhibitor has even a more deleterious effect. So this is telling us that if we play and help a little bit the epigenetic code, we can have higher rates of success, but still they are very low, like in the 9% from all the experiments. So this is the movie of how the nuclear transfer is done. Let me see if it opens.
This video was prepared by my colleagues Dieter Egli and Kevin Hagen. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out. And then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. This video was prepared by my colleagues Dieter Egli and Kevin Hagen.
Okay,
and
who is more technologically available here to do this thing? No, okay, come.
I think the problem.
In there. So let me open it and.
This video was prepared by my colleagues Dieter Egli and Kevin Hagen. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out. And then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. Drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part.
Okay. So
This
first
pipette,
they
you'll
will
know,
remove
isn't
the
really
nucleus
sharp
from
like a
this cell.
syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully into the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up.
And
So
then
that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again, a little hole, and the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of
That's
what's
how
called
the
somatic
nuclear
cell
transfer
nuclear
works.
transfer.
It looks very damaging, right? It's like they remove the nucleus, they just go to the cell, put it back inside, whatever was in the middle has to be regenerated again.
Very different from the text.
But but it works. It is a technology that works. Also, it's no. Okay. Okay, let's see again. And now in this one, no?
No.
No. This one. Go for it. Sadly. Okay, good.
There are, in the technique, there are two cylinders, one inside the other, and the other. I didn't understand, because then it approaches, it gets very close to the membrane, kind of forces it, and then suddenly we see, and and I kept thinking about like how mosquitoes do their thing, because they have like a duct, and then all the things that come inside it. And so
yeah, so it's a. This could also be why this is so inefficient, but no, they sometimes we protect the cell with some layer. So what you saw, there were like two layers, one of like a wall, and then the cell inside. And then they cross this and then really like push it physically through and insert things inside. And it breaks the membrane and it breaks many things, but the cells can regenerate very rapidly. I don't know how they see the nucleus to remove it, because I couldn't see anything, but if you are very experienced, you could do that. I did some, not in ovocytes, but we had a microscope where we can do microinjection of some things, some genes or some cells that were difficult to transfect, or sensors, and do live imaging. So you generate with a capillar a very, very, very thin like syringe, and then it has a force that it pushes it through hydraulic power, right? It just pushes it. How it doesn't come, and at the beginning when I was training, I will call this kill the cell game. If you do it too strong, the cell completely explodes. So you are training and it's like all your cells are just exploding. And then you understand a little bit with the experience how to really, how deep you should go, how much force you should use, and the focus is very important. In here, it's a movie and it's done very professionally, but when you are doing this kind of injections yourself, you have to be in the right plane, because these are three-dimensional and you see them in two-dimensional way. So when you push, sometimes you just break the capillar because you went too deep. Sometimes you use too much force and the cell exploded. So it requires like a lot of expertise. But once you learn to do it, you see that the cells have like breathe for a moment, like from the pressure, from the insertion of this thing. And then if they don't immediately die, usually they, it happens. I kill many cells in this way. They just like the membrane really forms again, but again, you are right that we could contaminate or we do some things, but these cells will probably die. So after, like in here, from the embryos created is not counting how many cells were injected and didn't survive the process. So this statistic is missing here. So many of the cells that were injected with a new nucleus didn't make it through. And okay, so we have very few clones, a lot of diseases, and one of the questions is, okay, we know that we have a barrier that comes with differentiation. Like as you said, we said the stem cells have a very open chromatin, and as we go along, they already have these domains, right? So maybe if we take as young as possible our donors, the efficiency will be greater. So this is what they did. They took they took nucleus from embryonic stem cells or from adult stem cells, fibroblasts, or different stages, And they saw that really influences, where is the statistic? Okay, here. How efficient it was. So for example, if we take from one oocyte to the other one, the efficiency is almost 100%. We can regenerate very, very easily with nuclear transfer. And then as the cells start a fertilized egg or 2-cell or 4-cell or already after birth or implantation, or a weaned animal, that is when they stop drinking milk and move to the regular food, the efficiency goes down. This is the SCNT, is the nuclear transfer. Some other are in vitro fertilization. So each process has a different efficiency, but the nuclear transfer, it's really affected by the age of the donor. And as we take them earlier, they are more similarly epigenetically, right? Because we are taking a nucleus in any case to the original one, so it's more easy to reprogram them and convince them that they are going to be a full organism right now. So this also has an influence on them. And also, when we analyze the gene expression of the cloned animals, they have a lot of genes that should not be expressed, but are expressed, meaning that when we reprogram them, we didn't reprogram them completely. We didn't erase all of the things that needed to be erased. So we have faulty gene expression, about 4 to 5% of all the genes. And the imprinted genes also don't know anymore what they were, like in the example with the gene from the mom and the dad, right? So in this case, they are getting the genes with the imprinted version, but they are also getting it as if it was a mom because it's the oocyte. So it's complicated. So they have like a confusion of what they are, and they don't get completely the full instructions of which genes to express and which genes they should be silencing. So for example, in here you see gene expression, and we have our control genes with high expression of this one, low expression of this one, so it doesn't matter the colors. Then we have the stem cells compared to the nuclear transfer, or the cumulus cells that are also a little bit more developed than the stem cells to the nuclear transfer. And still there are many, many differences. They are not completely like the stem cells or like the controls. They are different from them in many, many genes, telling us that this kind of reprogramming is still not perfect, and we don't get a perfect animal, and that's why they also get hidden defects, these cloned animals. So they can appear normal mainly when they are born, like some of them have defects when they are born, but some of them age earlier because they have accumulated damage. Many of the epigenetic modifications were not erased properly. So we already start with something that maybe is not as functional as really going through the whole process of zygote deletion of all the epigenetic modifications, renewal of the modifications, putting them in place as it should be when we clone from an adult nucleus. So these are also the degrees of abnormalities, and you can see that we have, with the age of the clones, the ones that survive are the most normal ones, but we have many, many, many defects, and they will die really early. And then the ones that survive and were able to make it to birth, then they start having some defects as they get older. So because of all the problems with the stem cells and with the nucleus and all these things, the induced pluripotent stem cells were invented. And the idea is that we can take an adult cell and then we can reprogram it to be a stem cell. And why can we do that? Because the genetic code is the same genetic code, but we just have to cross all these epigenetic barriers. So what happened is that the Yamanaka, that won the Nobel Prize, Gurdon won the Nobel Prize together with Yamanaka by the nuclear transfer, and Yamanaka for the IPS. So what he thought is like, okay, we already know from stem cells what transcription factors they are expressing. We know which one of them may be important. So let's start adding them into the cells. So at the beginning, he took 20 different transcription factors, and put them inside a fibroblast. So this is how a stem cell look like. This is not a single stem cell. Stem cells are really, really small and form these clusters. So we have here like a community of stem cells. These are how MEFs look like. So usually in the microscope, the stem cells will be really, really crowded, and it will look like this is a single cell, but it's a community. And MEFs already have like a cell body, like this is the nucleus, and then they have a shape, and they look, it's too small, I guess, for you to see, but they look completely different morphologically. But then when he started putting back transcription factors, he saw that morphologically he was getting something that looked more like the IPS. So at the beginning he put 20 transcription factors, and then he started saying, okay, do we need all of them, or we don't need all of them? So some of the genes that are known to be important for pluripotency are OCT, NANOG, and FBR15. And one of the things that changes is how they are methylated. So for example, in stem cells, they are not methylated. So they are, in here, each dot, a clear dot represents non-methylated site, and each black dot represents a methylated site. So stem cells are not methylated in these genes, and fibroblasts are methylated in these genes. So in these transcription factors that were added, 24, 150, 24, whatever, the different experiments, he managed to reprogram some of the genes, but not some others. Okay? Then he decided, okay, do we need all of them to form a stem cell? And start removing transcription factors. He will put 24 minus 1. So in each experiment, he could measure the efficiency and the colony numbers. So for example, if we remove this transcription factor, we get nothing. If we remove some other transcription factors, we have more efficiency. Then he did the same 10 factors minus 1, then 4 factors minus 1, and then he came to the conclusion that he needs 4 factors: MYC, KLF, SOX, and OCT. Okay, so it began not completely blind with hundreds of thousands of genes. It started with 24 transcription factors. And by transcription factors, I mean chromatin remodelers and factors that bind specific sequences that they knew were expressed in stem cells and were like highly expressed and unique to stem cells. So they came up with these genes, the Yamanaka factors, that if we put them inside the cells, we can reprogram our MEFs to be stem cells. And again, we can see in here, this is gene expression, this is qPCR. When we have a band, it means this gene is expressed. When we don't have a band, this gene is not expressed. So for example, these are stem cells and MEFs, our controls. This is how similar it is to a stem cell or how similar it is to a MEF. And start seeing with the different experiments what happens to them. So for example, in here, these cells are mostly like the stem cells, and they are different from the MEFs. So the origin was a MEF, a mouse embryonic fibroblast, but now their genetic profile or the gene expression is more similar to the stem cells. And also the methylation patterns become more similar to the stem cells. And then what we need to prove is that we need to prove that these stem cells can generate every tissue in the body. So first of all, again, the stem cells have some genetic profile, the IPS become more similar, and the MEFs are completely different in here. So the IPS still are not 100% like the stem cells, but are much more similar to the stem cells than to the MEFs. Now, the big test is, okay, we generated this synthetically in the lab. This is something that didn't exist in nature. Can this generate a full organism? And for this, there are two main experiments that are done. One, it's called the Teratoma Test. A teratoma is a type of cancer that includes, when we analyze it in the microscope and histologically, we see that it has all the layers, endoderm, exoderm, and mesoderm. So this is telling us that if we get a teratoma with all these layers, so this IPS can generate all of the different layers that are required for an organism. So this is what they did, and they got teratomas. And this is a nice proof, but still it's like a kind of an awkward way to prove that we can generate a whole new organism. So the second way to do is to generate a full animal. Let me see if I put these are the human IPS. Okay, so what they did is similar to what happens when we want to generate, in any case, a transgenic animal. I don't know if you know how we generate the animals that are knockout, knock-in, and all these things. Some of you know them. Okay, so we'll go. So in general, when we want to generate a transgenic animal, we take a stem cell. We put the gene that we want to put inside. Usually we add a puromycin or GFP or something that can tell me that it's inside. We also check that it got into the right sequence, that it didn't generate damage in other regions. So it's important for us to know. So for example, when we were generating the mice that I'm working with, the CIR6 knockout for the brain, we had to look at 800 different clones, stem cells, and only 4 of them were positive with the clone in the right side without other locations being inserted. So you also have to be lucky and also have to do many, many chances. So in this case, we put the gene that we need. In this case, what we get, instead of taking an ESL, we take an induced pluripotent stem cell. And then what we do also to identify them more easily is that our donor stem cell will be from a white mouse, for example, like this, and then the mom will be a black mouse. So we can see that we got something that will be mixed. Then we take a blastocyst that we were looking at them before, and we insert our cell in here. Now I have an embryo, something that will be an embryo that has mixed cells, right? Have the ones from the donor plus the cells that I want to insert, my IPS. And then this is given to the mother, the synthetic mother. And then we have pups. And these pups are chimeric animals. Most of the cells come from here, but some of the cells come from here. So I will have in a black mouse some white patches. And all our hopes go that some of them will get to the sperm. So in the next generation, we will have a mouse that is completely coming from the cells that we inserted in here, right? So we will have from maybe a black mother, a white mice that is completely white. So in this way, they generated mouse that came from IPS, giving the final example that really we can have a full animal that was generated from adult tissue, from a MEF, okay? And in humans, this barrier, if in mouse we have like some efficiency of 1%, 2% or even lower, in humans it's much more difficult. Something happened in evolution that is more difficult to generate IPS. The genome is more or less the same size, but we have additional barriers that we don't completely understand for the adult stem cells. And again, still we have some problems with gene expression that is not completely identical to what it should be. And just to summarize what is the criteria for a pluripotent stem cell, if we really manage to produce the IPS, is that they have to be immortal, but not transform, meaning they can self-renew forever, but they won't generate a cancer. Like in the experiments with the teratoma, it is in a specific mouse, a skid mouse with other conditions. They are undifferentiated, but can differentiate into different things. They have the clonality, they self-renew and produce more of themselves, and they can have all the developmental potential. They can become every cell in the body, maybe again not a trophoblast, but all the other tissue. So when we take this in tissue culture, we grow the cells and form embryoid bodies. These are already many, many stem cells, IPS, that divide together and form like a little body that looks like a very, very premature blastocyst. And then they can start differentiating spontaneously and give us different tissues. In vivo, we do the Teratoma test, or we produce animals in the germline. And then we have the different problems with the safety to generating IPS. Many people don't want to have anything that is transgenic, not because it's a big problem, we believe, but because the public is very afraid of everything that is transgenic. So also to improve the technology of the IPS, many safety methods have been developed. For example, the less safe are adenovirus. We put all the Yamanaka factors in a virus. It integrates in the genome. Well, the adenovirus doesn't integrate the genome. The lentivirus integrates to the genome. But then we can be causing damage, right, into our new cells. So all of these ones are efficient, but are less safe. And then what we can do is also episomal genes, like a plasmid. We put this inside, but then in cell division, it gets lost. So we needed it to form the IPS, but once they finish their role, we can get rid of them, and it's more safe. And one of the things that show to be the most effective is mRNA, as we are using now in the vaccines. Right now, all the vaccines are mRNA vaccines, or not all of them, but very efficient for corona, because we put the mRNA, we protect it with some chemical modification so it won't be degraded so easily. But after we highly express the gene of interest, it gets degraded, flushed out of the system, and generates very little damage. Same things were done with recombinant proteins, and we could say, okay, this should be the safest way, right? This is a protein, it has no genetic material, it cannot integrate to the genome, it cannot be reverse transcribed or whatever it is. But producing proteins that are functional is very difficult, because also the production itself generates a lot of aggregates, and also we have to clean this, and the cleaning process is also, sometimes we get a lot of debris, we get other proteins that were there. So in the end of the day, it's more expensive and it's less efficient than the mRNA. So nowadays, the most safe and targeting for humans are the use of mRNA. So this process, again, is very, very inefficient. And what happens is that we start the process, for example, to produce IPS, and they have different pathways that they could take to be IPS or to die on the way. And most of them, 90 to 95% of the cells, they don't make it through. They differentiate, or they differentiate into different things, or they just simply die. And some of them will manage to get to be a IPS. And what happens is that when we start the process, we have these cells that they will be in the end transformed into IPS, that we start expressing the mRNA of pluripotency. We are losing the MEF identity. We have metabolic changes and gain proliferation. So the first step is proliferation. The next step is we start producing microRNAs that block other things. Then we start with the histone modifications, so we start rearranging the chromatin, and then we have the DNA methylations that happen in the end of the process. The cells that don't manage to change, most of them, they just start expressing the mRNA, but they cannot go through the epigenetic modifications in the long term. So transcriptionally, they kind of started the process, but then they didn't go through the epigenetic changes that were required to really change the identity of the cell. So this is one of the things that blocks our production of IPS, that if we don't go through the epigenetic modifications, we won't get an IPS cell. And of course, now the IPS technology can give us, for transplantation and also for research. For example, we cannot, in neurodegeneration, we cannot take a biopsy from the brain and grow neurons from our patients. But we can take MEFs from the skin, not MEFs because mouse, but fibroblasts from the skin, and de-differentiate them and then re-differentiate them into neurons and try to understand what is wrong with this patient, what is different, without having to take brain tissue. So this also gives us a tool for research, give us a hope that we can put back some of the, if you have a mutation that can be corrected and then you get your own cells. And of course, it also helps us with the clinical trials. One of the main barriers we have when we are trying to discover new therapies and new drugs is that many things work in mice, but then when you try to go back to humans, it doesn't work. So one of the things that can help us check these things is to generate organoids. Organoids are small organs that we produce also from this IPS that we can make them to be more similar to human tissue and see if it's working. So this technology can also allow us to do these kind of things. So we are about to summarize this part, and I am not sure we will have time for the other part, so it doesn't matter. When we are at the top of this Waddington landscape, we can become almost anything. And this is possible, again, because we have the information and we have to decide how to use it. Then we start closing regions and opening new regions to become a differentiated cell with its own characteristics. And this barrier cannot be crossed without investing energy. And we can invest this energy in a synthetic way when we produce pluripotent stem cells by adding these transcription factors that change the epigenetic code, change everything in a synthetic way. So we can go back in this reprogramming to a state of pluripotency and then go back again to differentiate to anything that we need. And the main barriers here are, again, the epigenetic barriers plus accumulated damage. So many things are being tested, for example, as with the nuclear transfer, acetylase inhibitors or methylate inhibitors or activators or these things to improve the capacity of these cells to reprogram as precisely as possible, as similar to a zygote that went through all the process of deleting and reactivating genes that we should reactivate. And that's it. The other part was, I'll just show you, I think, one slide, so it won't be too long. Okay. These are so just for, how we study aging in general is that we also do comparative genomics and analysis of what is different in a long-lived species and a non-long-lived species. And very interestingly, humans, more or less, the average lifespan is 75 years. A tree can live for 5,000 years, but depending on who you ask, because many people say that the newer layers are alive, but the older ones are dead. So it's not exactly 5,000 years, but it depends who we are asking. And there is only one organism that we know that do not age. This is the hydra. It dies. It's not that it never dies. The population kind of maintains its own number, but they don't have any aging process. None of the things that we know that happen with aging, and it's probably because they have a huge pool of stem cells. So they have a very big regenerative capacity. And now there is also a lot of research in planaria. Because also planaria, if you cut them and you mix them and whatever, they will regenerate and form a new organism. So this is teaching us about the regenerative capacity and what can be done. So there are the different theories of why do we age, the antagonistic pleiotropic, the damage, and everything. But these are the main hallmarks of aging. One is genomic instability. This is the DNA damage, the acquired mutations. And then this also is with telomere attrition. And these are the drivers that lead to epigenetic modifications that also will start this epigenetic drift that will lead to the loss of proteostasis, all these proteins that aggregate or don't fold correctly, problems with nutrient sensing, mitochondrial dysfunction, senescence, and all the other things. So in my lab, I just want to show you that we try to understand what are the epigenetic changes that are leading to neurodegeneration. And what we do is we use genetic models of accelerated aging. I was very optimistic that I have so much time. Okay, so our main theory of why do we age is that we accumulate DNA damage. If we have damage and we repair, we recover. If we don't get the signal, we get cancer. If we have too much damage, we commit suicide and have apoptosis and tissue degeneration or senescence. So this starts a cascade of signaling that will lead to age-related diseases. And so in my lab, we study SIR6 that I told you a little bit before because it can repair the DNA damage. And also it regulates gene expression because it can deacetylate histones. So it has two main roles. Epigenetic regulation under normal conditions, but when things go wrong and there is broken DNA, it repairs the DNA. So when we delete it, the mice get accelerated aging. They look super old. They manage to live about 4 weeks of age with all the characteristics of being really, really old. If we overexpress these genes, mice can live longer, about 10 to 15% longer. So this is telling us that this is an important gene in longevity and in age-related diseases. And particularly in the brain, we see that there are, if we have inherited mutations in DNA repair, we will have microcephaly, ataxia, neurodegeneration, mental retardation. So many problems come especially to the brain if we don't manage to repair the damage. So also in Alzheimer's disease, Parkinson, ALS, all this neurodegeneration have increased damage in the brain. And the question is always, is this the cause or the consequence? And for this, we generated animal models that don't have SIR6 in the brain specifically. And then we can study what happens to these mice. So for example, these are the full body knockout, meaning they will die by 4 weeks of age, looking smaller with a damage in the back and loss of subcutaneous fat and colitis and all these things. If we only delete it from the brain, these mice become obese as they age without eating more. They are even hyperactive, they are more active, but they change also the metabolic signaling with the body. And if we check their learning impairments, mice, anyone has worked with mice? Also the molecular biology, no? So they are very curious, but of course we cannot ask them if they remember something, so we have to develop this kind of essays to learn about their behavior. So this is one example. We take a mouse and we put in a cage with two toys. So it has this circle and this cube, and it plays with both of them. But then we change one of the toys for this triangle. And then if he remembers that he already played with this one is a novelty, so he wants to spend more time with this kind of toy. So we can see how much, if a mice don't remember, they spend about the same amount of time with both of them. So this is one of the things that we can do. We have also the contextual fear conditioning, for example. As you have seen in nature, animals tend to freeze, right? When they are afraid, they just freeze. So if we put a mice in a cage and nothing bad has ever happened to him in this cage, he's not afraid and is a happy mouse in here. But if we induce an electric shock in the foot in this cage, it's a small one, but still it's a shock, it will be afraid every time I put him in this cage. But in a different cage, he's okay. But I can couple this with a signal, like this Pavlovian reflect, and then he will be afraid of this cage, but also if he hears the signal, and they should freeze. If we don't have SIR6 repair capacity and epigenetic regulation in the brain, these mice, they don't learn, they don't remember, they don't know that something bad happens to them in here. So through various of these kind of tests, we can test memory, like we have mazes and these things, and we can see how they are doing. So we know that if you don't manage to repair DNA damage and regulate epigenetic in the brain, you will start early neurodegeneration. By 3 months, they are already impaired. And if we take all the transcriptome of the brain of our mice and compare it in an unbiased manner to all the data that has been published, the top categories we get are neurodegenerative diseases. So we know now that we have a very good model for sporadic neurodegeneration. because most of the cases are age-related. 95% of the cases of Alzheimer are age-related, about 90 of Parkinson's disease, and most of them don't have any mutation. It's just aging process, and we know now that we can study neurodegeneration in our animal model because it's just very, very similar to what is happening in neurodegeneration. And in this way, we can see changes in brain morphology, in DNA damage signaling, in a cell death in the brain, and also neurodegenerative markers, for example, tau. Because, of course, if we don't have SIR6 and we have more damage, we can expect to have more dead neurons, right? But this is similar to neurodegeneration. And for this, we have to add the markers of neurodegeneration, and tau is one of them. So I'll just tell you that we also see reduced levels of SIR6 in Alzheimer patients. So we know that as we age, the levels of SIR6 start declining. And the levels of DNA damage start getting more, very high. So we don't have enough SIR6 to help us with the repair. And then if you are even more impaired with the expression of SIR6, then you cross this barrier of normal cognitive decline to a cognitive decline that is already pathological. So some of the projects in the lab are also trying to understand the promoter and the regulation of SIR6, what it gets silenced, right? We have it in our brain, it is there, all we need to do is express it, why is not being expressed? So we are trying to understand the promoter, the enhancer, what is changing as we age. Other things that we are trying to understand is how DNA damage changed all the epigenetic landscape. What happens to our genomes when we get damage in the long term? This is a constant damage accumulation, and then how this changes all our nuclear shape, because we are starting to lose also the nuclear membrane, like in laminopathies. This becomes weaker, and then the DNA becomes more loose or less regulated. So all of these things can help us understand why do we age, why do we get sick as we age, and also if there is something that we can do about it. So these are kind of things that we do in the lab, and of course I don't have to the 100,000 slides that I prepared for you, but this is just to give you an idea of what we do, and also we collaborate with doctors for human samples and all these things. So this is our field of expertise for those of you that are interested in going in molecular biology. That is super cool. But also we do a lot of analysis as well, because we need to combine all these layers of information that we get from sequencing and from metabolomics and proteomics and transcriptomics with behavior and all the other things to try to understand what is happening, particularly in the brain as we age, and how to keep us healthy, hopefully, one day. So just to finish, it has been really, really fun to teach you. I hope you learned some new things, and don't feel bad if not everything was clear. And feel free to contact me if you have questions and you need more explanations, or I don't know about reading material, because most of the things are from papers, and maybe it will be very complicated, but I can try to refer to some videos or explanations in other courses. And that's it, so thank you all for questions for this? No? Okay, sure.
I don't know if it's a question, but I was a bit shocked when you showed the use of iPS cells to check other diseases from other cells, body parts. How do you do that? Like, how is that possible? I would think that if I use the cell from another body part, I wouldn't be able to check mutations and the different things and to find the disease.
So what happens is that we have two main sources to produce iPS. One of them are skin and the other is blood. And we work with the lab in Boston that they generate an iPS library. They have more than 800 samples from different diseases, from humans. So what they do is they take this blood or skin, and then they produce this iPS. And now we have a bank from Alzheimer, Parkinson, and also from different patients, because maybe it's also a genetic interaction, because maybe you don't have a specific mutation, but you get two alleles from that together they don't work well, right? And then you get the disease. So also we may have a genetic cause for the disease that is not completely age-related or anything. So we get this IPS, and then you differentiate them to the tissue that you are most interested in. So for example, if you want to study the heart, and this person has an history of heart disease, but there is not known why, you differentiate the cells to cardiac tissue, and you see how they do compare to the library you generated from a person that has no heart disease, right? So we can start seeing if they develop similarly, if morphologically they have differences. In the gene expression, they are different. Or also in the epigenetic modifications, right? So we ignore the layer of the genetics, because this was not the part that we were interested in, and we check how they behave, how these cells become differentiated cells of the tissue we are interested. For example, one of the things that happen, if we take, you remember the laminopathy, the kids with the nucleus that is completely broken? If we take cells from these kids and produce IPS, the IPS look normal. They look okay. When you start differentiating them, they become sick again. So it's something in the maturation of these cells that gives them the disease, right? In the beginning, they were okay. So when we take from patient cells and put them as IPS, they may seem still normal. But we know that this patient specifically has neurodegeneration, but doesn't have cardiac problems. So if I differentiate to neurons, I will start seeing differences between them that I don't see in the IPS or any other tissue. So this is how we try to understand better the specific tissue, particularly the ones that we cannot grow now from the person and take a sample. Okay. There was one question? Yeah, sure.
So for cloning, when I think in the presentation you said that the first clone that was ever successfully produced was in 1952 a frog or a tadpole,
yes,
something like that, right?
In amphibians, but easier.
Yes, but then the dolly, which was the first mammalian, was in the 80s, right? If I remember.
I think in the 80s.
So, and just looking at that video, it looks like a very simple procedure. You just take the thing and put it back. Like, what was the technological development?
So
what were the difficulties? Is it just ethics?
It was also ethics. It was also that really the frogs' ovocytes are very, very big, that you don't even need a microscope to see them. And in many cases, many of the discoveries also in the cell cycle and also in electrophysiology and many things were done in this because they are so big you can see them and then you can manipulate them more easily. And the other ovocytes are much smaller, and the technologies and the microscope also need to develop, because really there is a huge difference between the cell that you can see with your naked eye or just a binocular, or these methods where you also have to maintain this nucleus in this cell in size and come with the microscope and inject these things. So even if it looks very simple in an hydraulic kind of thinking, it requires a good microscope because, as I told you, the focus is very important. It requires the technology also to implant this back again in the animal. And this also took time and many experiments to prevent infections and to know how to prepare what is the name? The adoptive mother, right? You have to put it in a mom that was not the real mom and she's not really pregnant, so you have to make her believe she's pregnant. So also the development of these techniques from amphibians with these eggs to mammals had like a very long learning period, also to develop better techniques for the nuclear transfer and also to learn how not to kill the mom, to get the right signals, when it should be implanted, when is the last time that you can implant this. So I guess it took also barriers, ethical barriers, and also technological barriers that now we think, oh, we have the microscope and they all work. Well, they didn't have this kind of microscopes in the 50s. Okay, any more questions? Okay, so you are free for the coffee, and okay, sure. Who will take the picture for us? Okay.
Transferencia de núcleo, lo que es, no sé si sabes sobre lo del mamut que quieren como
Sí, que querían clonar. La idea era ahí como hacer como un passage a elefantes, ¿no? Como lo que yo